I think we can get started. So welcome to the lunch and presentation. I hope you enjoy lunch. I will maybe, whenever there's a highlight, I might quickly tell you, and we make sure that everyone gets the most important topics of my presentation. So my name is Raphael. I'm taking care of product management, marketing, and sales at a company called c-LEcta. We are certainly well known for DENARASE, the GMP endonuclease, used in lots of viral vector manufacturing processes. I have a scientific background. Before I joined c-LEcta, I used to work at companies like 𝗞𝗲𝗍𝗄, AstraZeneca, as well as Novartis. And what I would like to discuss with you today is how we engineer DENARASE towards our new product, DENARASE High Salt, that allows for highly efficient DNA removal in viral vector manufacturing processes, independent of salt and pH changes. So just a few words about the company behind DENARASE, c-LEcta. We are a company based in Germany, Leipzig, and we were founded in 2004. So actually in 1 month from now, we are celebrating 20 years. If you're around in Leipzig, come by. We have a big celebration happening. And all the enzyme engineering that we do as well is based on a key technology that we have, a key enzyme engineering technology, allowing us to engineer and optimize enzyme in a very fast and efficient manner. And yeah, in the meantime, we established ourselves as a supplier of high quality R&D to GMP grade enzymes to the pharmaceutical industry. And we also have quite some products in development. So if you would like to improve an enzyme in some of your manufacturing processes, don't hesitate to reach out to us. We are very much used to work together in a collaborative business model so that an engineered enzyme makes it into pharmaceutical processes. So let's get started. Endonucleases. Most of you in the room might be using them in some of your viral vector manufacturing processes. And of course, it's used to degrade unwanted residual DNA during purification. And endonucleases have been used since more than 40 years, and it actually mostly started with vaccines, being it vector or conjugate vaccines, where they also had to remove host cell DNA. And in the last 15 years, with the evolution of cell and gene therapy, of course, the use of endonucleases was growing significantly. And the main background why endonucleases are used and DNA has to be removed is certainly regulatory requirements. So we have to make sure that the amount of residual DNA in your drug is below a certain dose of at least 10 nanogram per dose. And you also have to make sure that any remaining fragment is below a 200 base pair size in order to limit the risk of your drug. And that actually led to a pretty standard workflow on how endonucleases are used in viral vector manufacturing. So you have viral vector being expressed, you go over to harvest, you might do a cell lysis step depending on the process, and then immediately afterwards, you normally treat or you normally add endonuclease. The solution might still be warm from the expression, it might cool down to room temperature, and that's how it's normally done. It's continued by purification, and finally you're ending up with a drug, a vaccine that can be administered to patients. Standard workflow. The enzyme has also been pretty much a standard. So most processes do and still do rely on an endonuclease, a wild-type endonuclease derived from a gram-negative bacterium called Serratia malsescens. So that has been the workhorse in the last 40 years. Also, our DENARASE is a wild-type Serratia malsescens enzyme, and it's available since 2012. We have it from R&D up to GMP grade, representing the industry standard. In the meantime, more than 250 customers rely on it. Most of the big pharmaceutical companies also qualified DENARASE to be used in their processes, and that led to the fact that for us as a rather small company, led to the fact that DENARASE is applied already in commercial and hundreds of clinical and preclinical manufacturing processes. So for us, it's great to see the widespread use of the enzyme. However, we also ask ourselves, we have more than 60 to 70% of our employees being scientists in the lab. We ask ourselves, what can we optimize? How can we help with an optimized enzyme that your processes are running better? And there was one aspect that has been discussed in the industry for quite some time. That's about salt. High salt conditions, high salt concentrations, or I would even say salt concentrations that can be adjusted to your process, to your viral vector. It can really help because it can help you to reduce the formation of aggregates of your viral vector, and it also can help to make residual DNA that we want to get rid of more accessible for degradation. And ideally, that will help you to increase the stability of the viral vector. It will help you to increase the yield of the viral vector and ultimately meet your targets, your requirements regarding purity and safety. There is one drawback with the standard 𝗟𝗲𝗿𝗀𝗃 endonucleases that most of the people are aware of. With increasing salt concentrations, you certainly see that the activity of a normal 𝗟𝗲𝗿𝗀𝗃 enzyme, such as DENARASE, is increasing, as shown by the yellow line. And there are also a couple of endonucleases on the market that are called salt-active or salt-tolerant endonucleases. We now abbreviate it in this presentation with SAN. And most of them do have a strong dependency on sodium chloride concentration. So the higher you go, in this example, the better activity you have, but you might optimize your process for the enzyme and not for your viral vector or for your preferred process conditions, as well as pH might also be not ideal. So we started working on our normal DENARASE and thought, can we engineer this enzyme? Can we take wild-type 𝗟𝗲𝗿𝗀𝗃 endonuclease and engineer it that it actually fulfills the requirements of high or medium adjustable sodium chloride concentration as well as pH? And we asked quite some of you. We discussed with more than 40 companies being active in the viral vector field on what their preferable conditions are. So you might think, oh, it would be great to have an enzyme that has strongly increasing activity at rather very high sodium chloride concentrations. You might also say, ooh, I would prefer some kind of a peak-like behavior at a certain sodium chloride concentration. Overall, what we concluded from an extensive interaction with viral vector manufacturers is that when it comes to salt, when it comes to sodium chloride, it's very important that as a broad spectrum of sodium chloride can be covered by one enzyme, ideally 100 to 500 millimolar of sodium chloride. If the enzyme works reliably under all these concentrations, that would be great. And something that wasn't discussed too much in the past was also pH. You might be aware, most of the endonucleases, when you look at the specification, when you look at the certificate of analysis, you see that release conditions are mostly done at pH 8 or even higher. And your process might not be running at that pH, but rather at a more physiological pH of 7.4. So we also took attention when developing the enzyme that pH 7.4 is that the enzyme works under these conditions. So we started working on it. we introduced single mutations into the enzyme and combined it to several mutations in one's enzyme, and we tested for improved activity at 500 millimolar sodium chloride. And we always compared, of course, the activity to wild-type 𝗟𝗲𝗏 endonuclease or wild-type DENARASE. And luckily, we found quite some interesting mutants of DENARASE that had activity over broad salt concentrations and also activity at process-relevant pH. What I will show you in the next couple of slides is technical data comparing or showing behavior of the enzyme. Oftentimes, I will show the activity at pH 8, as shown here on the left, and activity at pH 7.4. The background is release conditions. What you read on the specification of most suppliers is done at pH 8 or higher. However, most processes might prefer to run or to use the enzyme at a pH of 7.4. What you can see here is 3 variants that we selected and that we tested further for activity. You can see a pretty nice and stable activity over the whole range from 100 or from 0 to 500 sodium chloride concentration of sodium chloride. We finally decided to go with variant 3. And indeed, after a lot of extensive additional testing, this is now DENARASE High Salt that you can purchase or that you can get a sample for testing. Question might arise, okay, when shall I use DENARASE? When shall I use DENARASE High Salt? The two enzymes are actually pretty similar. We only introduced very few mutations into the enzyme. So quite some characteristics are similar. However, there are some aspects that are different in order to make it work at elevated salt concentrations and pH. First of all, enzyme origin. DENARASE is wild-type 𝗟𝗏𝗏. High salt is engineered. Production host, in our case, is a Bacillus species. This is a specific species that we are able to produce in because we have intellectual property on it. It allows to high titers, and it's a gram-positive strain, so reduced risk of endotoxin in our process. Molecular weight is almost similar. Just a few mutations do not change it. Temperature optimum is the same. pH range is more or less similar. What slightly changed is actually the amount of magnesium you need. As you might all know, magnesium is a cofactor. Without magnesium, the enzyme is not going to work. For DENARASE High Salt, it's beneficial to have slightly higher magnesium chloride concentration in your solution, and it increases activity, but the enzyme works at 2 to 25 millimolar. As well as the isoelectric point slightly changed, mostly due to the fact that we changed some amino acids on the surface of the enzyme. And so far, what we hear from companies applying the enzyme already, it's easily possible to remove it during purification, which is one major question that is asked. So, to maybe even make it a bit simpler, when to use DENARASE or DENARASE High Salt, we also have a rough rule of thumb. So what we did is the slide. Okay, you can read it, but the slide somehow changed the format a bit. So we did, with DENARASE and DENARASE High Salt, we did a endonuclease activity assay, as we do in our quality control labs. And again, we show here process relevant pH 7.4 conditions and QC release relevant pH 8 conditions. Again, you can see at pH 7.4, DENARASE High Salt shows very nice behavior over the whole sodium chloride range, whereas normal DENARASE certainly has a decreasing activity the higher you go with sodium chloride. At pH 8, we generally see slightly higher activity. However, if you test it in your process in your lab, you will see pH 7 is the enzyme is super active, you will not need to adjust the pH too much in order to make it work. Recommended usage, as a rule of thumb, from 0 to 200 millimolar salt. It's just perfectly fine to, in most cases, use normal DENARASE. It's a cost-efficient way to do it. If you go higher, I really recommend to use DENARASE High Salt. If you just want to have one enzyme in your lab for all the processes, go with DENARASE High Salt. So in summary, we can say DENARASE High Salt has excellent performance under elevated salt conditions and also works well at over a wide pH range. And of course, we have much, much more data. If you're interested in additional technical data, we have a whole validation guide that can help you looking into the details. And we believe that these two enzymes together, DENARASE and High Salt, they really are unmatched in terms of versatility and efficiency for your processes. And especially DENARASE High Salt has been designed, has been engineered to fit your process. So ideally, you don't need to adjust your process to the enzyme, but you take your favorable conditions in terms of sodium chloride, in terms of pH, in case you have other factors, and the enzyme will do a great job. How does it compare with other commercially available endonucleases, commercially available salt-active endonucleases? We did an internal comparison study, so that's lab work on our ends. We tested 3 commercially available salt-active nucleases, abbreviated here with SAN1, SAN2, and SAN3, and we compare it with DENARASE High Salt. Again, here you see pH 7.4, pH 8, mostly for the process. What is the most relevant pH is maybe on the left, pH 7.4, and you can see over the whole spectrum, over the whole salt concentration, DENARASE High Salt had by far the highest activity. So again, hopefully this can support you in your process so that you don't need to do too many adjustments, but rather use the enzyme and it works. Again here, generally, most enzymes are slightly more active at pH 8, but DENARASE High Salt works very well at lower pH too. Even if you go below 7.4, you still see way enough activity. At this point in time, we certainly, when we got this data, we certainly were very interested. How does it work in a real process? We don't have a viral vector lab in our company, so we started collaborating early on with quite a couple of companies, and we started to collaborating closer with the cell and gene therapy Catapult, an organization in UK that does a lot of process development and R&D for especially small to mid-sized viral vector companies. So they have a huge knowledge on different processes, and they did a benchmarking study. They used their common AAV2 platform process. They generated lysate, and then they tested DENARASE High Salt and compared it to normal DENARASE and SAN-HQ. And what they tested in the first attempt is simple viral sealed by capsid ELISA. So how does increasing salt and the presence of enzyme of the different enzymes have an effect on viral sealed? And secondly, DNA removal, very important. They had a good picogreen assay for that. And this is some of the early phase. These are some of the early phase results they got. When they looked at the viral particle yield, you see it on the left. Indeed, they were able to see a slight increase with increasing salt. You can see we started with pH 7 and added increasing amounts of sodium chloride. There's an increase in yield. However, I'm pretty sure if you tested with a less robust AAV vector, so this has been a platform AAV2 vector, pretty stable, pretty reproducible. You might even see a better increase in yield. We have to say with SAN-HQ, there were slightly fluctuating results, mostly due to the fact that pH 7.4 and the rather low amount of the enzyme that was added was maybe below the ideal performance of the enzyme. So we added 50 units per ml for all 3 enzymes. And what was really remarkable is, is the effect on DNA removal. As you can see, DENARASE High Salt was by far reducing the amount of double-stranded DNA the most. And more interestingly, it didn't matter too much if you had 350 millimolar sodium chloride or 500 millimolar sodium chloride present, or it didn't matter too much if you increased the pH to 8. It did a very efficient and reproducible job in reducing DNA. So we can conclude with the data we got from collaborator Catapult. DENARASE High Salt removes DNA from AAV2 lysates with highest efficiency. It outperforms regarding extent and also reproducibility of DNA reduction. And it worked very well under process or production relevant pH and salt conditions. So with that, I'm coming to the end. In summary, DENARASE High Salt, we believe it's the industry-leading enzyme in terms of performance for DNA removal. It's highly active at process-relevant conditions. It's tailored to fit your process. So you just need to add it, and hopefully it's doing its job. I'm pretty convinced. And last but not least, DENARASE High Salt is compatible with the normal DENARASE ELISA kit. It's just a few mutations that we introduced, and you can still detect and quantify it with a normal ELISA kit that is available, meaning that only one assay that needs to be set up in your QC, in your process development labs. You don't need to have several ELISA kits. And together with DENARASE, which we consider to be the most cost-efficient GMP endonuclease for low salt processes, we believe we have a complete toolkit for removal of any residual DNA in your viral vector manufacturing processes. If you're curious, you want to test it in some of your processes, let us know. Just scan all the samples. You can have a sample of DENARASE High Salt or DENARASE or both together. We have a lot of additional data from relevant process or process-relevant data from Catapult that is also presented in our webinar next week on the 27th of August. So maybe you might register for that one. If you want to talk to us during this conference, we have a booth 507, and on Wednesday and Thursday, we're also going to present posters. So with that, I hope you enjoyed lunch and it was entertaining and interesting. I'm happy to take any questions.
Thank you for entertaining us during lunch. That was great. You mentioned that removing the DENARASE High Salt is something people are concerned about, but you said it's straightforward. Could you speak to that a little bit more?
Yes. So with normal ribosome-derived endonucleases, I think removal simply works. So you have it present during the treatment step, and afterwards you do chromatography steps such as capture and additional chromatography steps. And normally it just flows through in some of these processes. And the latest, when you polish, the enzyme is gone. And so far, we didn't see any difference when customers tested DENARASE High Salt in their processes. But of course, we have much less knowledge yet. This enzyme has been launched in March. Yeah. But I'm curious to hear, and we can also give you samples for testing in your process. Can you maybe take, I think the microphone doesn't work. Sorry. Hopefully it works now. Not really. Sorry. Maybe you can. Sorry for that.
Very nice. Have you
perfect.
compared the performance of the DNARASE high salt with the 𝗲𝗲𝗃𝗃 newest benzonase high salt, salt-tolerant?
Yes. We just, so it was launched fairly recently, and we just got some samples, and so far we don't see a difference in terms of, let's say, that DENARASE High Salt works by far the best.
Anybody else? All right. Thanks, Raphael, for a great talk.
Thank you.
And we'll close this session.
Thanks.